Okay, so it's my pleasure to introduce Bill Idsardi today for today's seminar. He completed his dissertation on the computation of prosody at MIT under one of the founding fathers of modern generative phonology, Morris Holley. Currently, Bill is professor and chair of the Department of Linguistics at the University of Maryland. And Bill's research largely centers around how humans perceive, produce, and process speech. He has relentlessly studied these questions from a transdisciplinary perspective, including signal processing, speech perception, phonology, and neuroscience, all the while fully integrating computational and experimental methods and insights into his approaches. So without further ado, I'll introduce you. He can help talk today about representations and computations for serial order. And I'd just like to add that it's been a lot of fun working with Bill for the past decade or so. Please give him a warm welcome.
Thank you very much, Jeff, and it's great to be here. So this is not going to be a very complicated question, and I hope it has a simple answer, too. But I think many of the questions that are not complicated but fundamental take us a long time to answer, and this is one of them. So there's the abstract, which you sent out before. Since this is also framing what I do generally, I'll just say something briefly about how I approach problems. So my general approach to problems is one in which we have a problem of finding really what the fundamental objects are and their relationships. And so that, from a computational perspective, is just to say, what are the data structures? What are the data relations? What are the algorithms that underlie the solution to a problem? And the problem that I've been looking at all these years has been human speech. But you could take this same perspective on any other problem. Now, in the case of language, this is involving at least mappings between three very large systems, which are the memory system, which we know remarkably little about, actually, in human beings. The action system, production. I don't do a lot of work on speech production, but this informs some of the answers to the question. And speech perception, that is audition, or auditory neuroscience. We know quite a lot about that, mostly from animal models. And then we try to import those into human beings. So the other touchstone for me, really, in addition to people like Morris Holley, has been David Marr, who died very young. But he had a very influential view about how you should do the connections across these things. So he famously posited three levels of description or explanation for these problems. A computational level, where you formulate the problem statement in as precise a way as you can. The other level that was there before Marr was the implementational level. If you tear apart the animal, how is it actually doing it? His real contribution was this middle level of an algorithmic level that tries to connect the computational level with the implementational level by providing a concrete example of a solution to the problem, where you spell out what the data structures and algorithms are. Now, in all of these cases, you can find slippage between these two things, so your algorithmic solution might not quite solve the problem you were after, and the implementations might not quite implement the algorithms that you were after. So there might be some limitations, say memory limitations, such that the implementational level is not a full implementation of the algorithm. So answering these questions and providing the linking hypotheses between these two things, I think, is the very important part of my work. And this necessarily involves collaboration. Really, at this point, I have hardly any single authored publications in the last 15 years. And I'm proud of that fact. So, I know there are other ways to work, but I love working with people. And one of the things that we found out, I think, is that many of these answers require only restricted models of computation. That is, you don't need a full Turing machine to solve the problem. And so that's also going to play a role here. In language, when we look at language, ultimately we're looking at form-meaning pairs. Everything I'm going to talk about today is going to be on the form side. I have nothing to say about meaning today. In particular, what we look at is this memory action perception loop that we have, where, for instance, if you want to say a word, you would draw that word out of some long-term memory store, and you would somehow convert that into a pattern of motions that you can do with your mouth, which then cause vibrations in the air, which you can then perceive at your ear. The problem is, much of the time, is that we're restricted, and this is this sort of problem of looking inside things from science generally, to an external physical form. So we can measure it with a microphone and then try to back infer what actions must have taken place. We can manipulate the physical form and see if we get different perceptions. But the box that's really hard in here is the memory box. We don't have any way at the moment in human beings of directly doing this, although you can see in mice now, if you're following science, they've been able to implant memories in mice at this point, mostly having to do with grid cells, so knowledge of place, which provide one method of trying to think about the problem. The last thing was this work that I had done with Jeff, which suggests that really we're looking at very restricted computational power to solve the problems that we've got inside phonology. If the outside circle is Turing machine complexity, which it is, then we're looking down very far inside this to find answers to our problems in speech. Okay, so an inspirational story that I'm going to tell you now, which is not my work, but work by a person who's no longer alive and has not been alive for many years, and a person who is very much alive and is my colleague at Maryland, Catherine Carr. So the inspirational story here is also one from audition, but it's a simpler problem than speech. It's a problem of locating an object given its sound. So if you have a sound, then you know where that is relative to your body because you have two ears. Many animals have two ears. There are other ways to solve this problem. You can do superhuman accuracy with this if you have a microphone array. So if you allow yourself thousands of microphones, you can solve this problem better than animals can solve this problem. But we've been provided with two ears, and so we can solve this problem by looking at what is called the interoral time delay. So the delay in the arrival times at the two ears. And if you know, as the animal, how far apart your ears are, then you can use the time of arrival just to physically calculate what the angle must be. So this is a relatively simple situation physically. So the physics of the situation in this case, the physics maps the position of the sound to the delay at the ears. That's just part of physics. And then that is mapping essentially position in space to time inside the brain. So you're going to have to undo that. You're going to have a difference in time in the brain and back infer what that time difference meant in terms of a position. So this was, so there's the geometry of the situation. An algorithmic perspective on this, adapting the vocabulary from Gallistel and King's book, would be something like this. For event E, record the time of arrival at the left ear. For somehow figuring out that this is somehow supposed to be the same event. For event E, record the time of arrival at the right ear, calculate the time difference by doing a subtraction, and then use your trigonometric functions like arcsine to figure out where in the field that was. Now, that taken at face value seems to mean that we need a central clock that is governing the time, like so that you can't have lots of clocks in the brain. You have to have like a clock devoted to this that both ears can have access to. You have to have memory registers because you have to be able to store these values, like the time, to take a timestamp, store that in a register. You have to do addition, multiplication, you have to do trig functions, you have to do the inverses of all those things, right? And that sounds like a lot for the animal to have. And in fact, that's not at all how this problem is solved in animals. Even though that's a good perspective on it, we won't see any of those things. We won't see a central clock, we won't see any of those items. So in fact, like this is also well known in other areas. So catching a fly ball would seem to involve lots of notions of trigonometric functions, but in fact you can do it by simply moving so that the ball stays stationary in your visual field. So if it's rising in your visual field, you should move slower. If it's falling in your visual field, you should move faster. If you only have one eye, that's more difficult. Here is Jeffress's answer in 1948. He guessed the answer, and then we found that he guessed right, at least in some animals. And so this is known as a place theory of sound localization, and basically all it is a bucket brigade. And you do this bucket brigade by just wiring longer and longer wires. So the left auditory system, the left ear sends a signal into a bank on the left-hand side and into a bank on the right-hand side. And all you're doing is looking for the coincidence. And so the coincidence detectors are located along those fibers. And when you have the coincidence of two signals coming in from the right and left ear, they will coincide at some time delay. So each one of those dots represents a time delay of a certain amount, and then that maps to the position. Right? So it pre-calculates all the possible time delays that it can detect. Right? So it has a definite notion of just noticeable difference, which is physically instantiated in this. So it's not perfectly accurate, but it's good enough to tell you about 1 degree of arc, actually, in the auditory field. So that was the idea, was that it would wire up like that, like a bucket brigade, and coincidence detectors were the method for finding this. 40 years later, 40 years after Jeffress had suggested this, Catherine Carr published in a number of places a verification of this in birds. So the top one is from chickens. That wasn't her first bird, actually. Her first work was on barn owls. And if you want to see Catherine, she teaches every summer at Cold Spring Harbor in the neuroscience program there. So her first work was in owls, but it's easier to see in the chickens. So the chickens lay this out just directly. And so they have exactly what Jeffress predicted. Now it turns out that, so that's a place code. The owls have a multi-place code, so they have the same code recapitulated over and over again. So just, you know, copy and not modify in this case. And then finally, in mammals like gerbils, it's a more complicated code that we don't fully understand yet, so there's still work to be done here, which is a population rate code. So in fact, the medial superior olive has a bunch of cells that respond differentially in toto, and the more off-axis it is, the more they fire. So like that's a different code. So you can see here that there are two implementations in different brains that cross. Mammals have one system, birds have another system. They both solve the problem. They seem to be solving the problem in the same way. They calculate the interoral time difference, but they do have different implementations. So this recapitulates what Marr had said. And so in this case, in this story, we have really a story from soup to nuts, as my former colleague Amy Weinberg liked to say. So we have a computational theory based on the physics of the situation. We have an algorithm for mapping time delay to angle in general, and then we have implementations either as place codes or rate codes. This is what we'd like a story of this detail for how you recognize, say, human speech sounds. This is what we're after. And the problem that I'm going to look at today is just if you have a sound and then another sound, how do you represent that? Like, so how do you go, if you have A, B as a sequence, how do you do that? How do you represent that? And there are many potential solutions here. So just to situate ourselves, here is what we know about acoustic sequence perception. This is a one-slide summary of a book by Richard Warren. And so music in general goes about 2 per second. So 120 beats per minute is a very common tempo for music. And so each note is taking about 500 milliseconds in general. Speech is, from that perspective, about twice as fast in general. So the syllable rate in speech for many languages, in fact, like really all of the languages in the world, is about 4 to 5 syllables per second. So that's about the rate that it's coming in. This translates to about 10 to 15 speech sounds per second, because you can have more than one speech sound in a syllable. And so this is intelligible, in fact, even if you double that rate. So you can speed up your audiobook. You know, there's a little thing on the audio thing to run it a little faster, but you can't run it 10 times faster. You can run it a little bit faster and it stays intelligible. So you can get to about 30 phonemes per second without too much trouble. So the 2-element sequence pairs, just in general, telling A, B from BA for any acoustic signal is about 20 milliseconds of duration in A and B. So you're very good at this, like the threshold of detection. The threshold for telling whether two things are synchronized or not, like whether two sounds are absolute synchrony, is even better than that, 2 milliseconds. But to tell A, B from BA, you need about 20 milliseconds of sound in A, 20 milliseconds of sound in B. But as Warren says, the initial and terminal sounds of sequences, and this should come as no surprise to anybody who's had a moment of psychology, that the first thing in the sequence and the last thing in the sequence are special, is definitely true in this case. So when you try to do longer sequences, so when you try to do acoustic sequences of buzzes and hisses and whatever, A, B, C, D versus A, C, B, D, now the things, the A's, the B's, the C's, the D's, need to be 200 milliseconds long for us to recognize the order. And so this is a syllable duration, not like a segment duration. So in this case, speech is a huge exception here. Spot, stop, pots, pops, ops, and then even the post and toss, these nonsense. You can tell those apart, even though those are going much faster than 200 milliseconds. In fact, the total duration for spot is about 200 milliseconds. So in this case, we have at least one example of a case where speech is special. It's somehow better, you're better at doing the order in speech than you are of acoustic sequences in general. So there must be something special going on there. Exactly what, we don't know. And so the view on sequences, we can take three views here pretty quickly. So we'll take a view from computer science, which will sound a lot like the Gallistel and King view. And then we will take a view from psychology, which has come up with extremely creative solutions to this problem. Like things I never would have thought of. And then we'll come back and sort of say, what do we need to know from linguistics? So strings in computer science, which are the representation for sequences of this sort, are usually just done in, say, a block of memory where the adjacent cells of memory hold the content. So in this case, different colored squares. Another way to do this, which is more flexible, is what's called a linked list, where each one points to the next one in sequence. Now this is also well known from psychology as an associative chain. So this one is chained to this one, is chained to this one, is chained to that one. And most of the work in computer science then goes about to try and elaborate these data structures in various ways so that we can get extensions to these. This is a doubly linked list, so you can go both forward and backward in the list. And then our questions about these are generally, how efficient are they? And so for this, we usually have a bunch of operations that we're interested in. How long does it take to concatenate strings? So I've used spelling here so that it's more accessible to everybody. So if we have dog and we want to make it plural, we add something onto the end of it. In the spelling system, that's an S. So we've concatenated those two things. Sometimes, even with that operation, we need to insert something extra. So the right form for boxes includes an E that has no extra value to the thing. It doesn't mean anything. So box plus S in the plural, then we have an extra symbol there. Same thing is true for thermometer. The O, Mark can tell you more about this if you want to know. The O in thermometer doesn't mean anything, but it's there for some reason. And then we can have other things like deletions. So in this case, from the spelling, in baking, we drop the E and add ing. I'm sure you heard that in second grade. And in waitress, we drop the E when we add ESS that you probably didn't hear in second grade. And you have other sorts of substitution. And then our evaluation of the data structures is how efficient and effective are they at doing all of these different computations. And you might choose one data structure over another because it has an efficient implementation of, let's say, concatenation, if that's what you're going to do a lot of. Okay, so that's sort of the perspective from computer science. The perspective from neuropsychology is different. I don't know that vectors play any place. I mean, the place codes, I suppose, could be understood as vectors, that sort of in-place memory locations, 1, 2, 3, 4. It's true that place codes have adjacent cells, but the cells cannot hold arbitrary symbols. They can only hold that thing that we computed that said, this was a coincidence of the left ear and the right ear responding. Right? So in computer science, it's typically the case that we believe that these memory cells can hold, maybe they're restricted to a type, but they can generally hold any contents that we want to put in them. As I said before, linked lists look like the psychological version of associative chains. And there's a quote from Lashley that I skipped for time. These do occur in animals, these chains. So they're called reflex chains, or a modern version of these are synfire chains in neuroscience. Mostly in locomotor function. And the best one of these, I think, is this experiment that was done at the beginning of the neuroscience tradition where they took an earthworm and chopped it up and then connected the segments by small pieces of string. And so as the front segment moved, it pulled at the next segment, which then moved, which pulled at the next segment, which then moved, and so on. That's one sort of reflex chain. And so it's not the case that it's just peristaltic movement in the earthworm. It's actually the fact that the segment is moving forward, pulling on the next segment, is this external stimulus from the standpoint of the second segment that causes the reflex to go. So there are different ways this can work. Synfire chains are internal, they don't require that. And so in this case, instead of putting something into a memory slot, really we have relations between memory type cells. This is very different. So the version from computer science is we take this thing and we just plop it in there. And these things you have to pre-calculate an awful lot of stuff. You have to pre-calculate most of the things you think you're going to see. And so Lashley had an objection to these sort of associative chain models from speech. And his intuition, even though he wasn't trained as a linguist, was that you could have things like right, and you could have things like tire, which I can't quite pronounce the way that Lashley did. So for me, they're not perfect anagrams of one another. But we did have some before, spot and tops and so on, where the order of the elements is not predetermined by the nature of the elements themselves. That is, when you say an R, you don't have to then say I. You can say any other thing you want to, like, you know, whatever is valid in the language. So you have to have a flexible notion of sequence in this case. So it can't just be the way it is where in the one segment of the body, if that moves, then it's the next segment of the body that moves in all cases. So this, we need a much more flexible thing than that Synfire chain. And so there have been several models proposed in psychology. One is a positional coding model, where you say, I saw this thing in this position. So you pre-calculate all these things. So we limit ourselves to a certain length of string, say 4 in this case. And then we say, I will give you a unit that means S in position 1, or S in position 2, or S in position 3, or S in position 4. So we would code spot as we saw S in position 1, P in position 2, O in position 3, and T in position 4. So the objection to this model, which has mostly come from visual word recognition studies, has been that this has no unified concept of T. It only has T at a position. Now, so you need to augment these models in some way to get that notion of a T anywhere in the string, right? But the fact is that they have lots of work from visual word recognition showing that you do know that, and so these need to be augmented in some way. These are also very popular in neuroscience. Here's one from Eichenbaum, who's like the major figure in memory at this point in neuroscience. And so he has a review article in 2014 on memory in mice for place cells mostly. And so there's his temporal context model, which is that one. And the chaining model, in this paper, he suggests putting those two things together to give a chaining model and a temporal context model. So you could, this is not to go into this in any detail, but just to let you go and look at that if you'd like to. So that's sort of state of the art in neuroscience thinking about this. There are other models out there. So another major series of models in psychology are called activation models. And so in this case, the relative activation codes for relative position. So if you have more activation, you're closer to the beginning. Or if you have more activation, you're closer to the end. You can do it either way. This is also known as competitive cueing. There is neural evidence for such codes in monkeys for motions, for hand motions in monkeys, where they're trained on. This is recent evidence in the past 2 or 3 years. And this works reasonably well as a coding scheme for a limited number of items, like say 5 to 7 items. The real problem with these models is the repetition of an item. And so in this case, in psychology, with the visual word recognition models, the usual answer is to consider only words that don't have repeated letters. Not repeated locally, but repeated at any distance. So like this is a very strange sort of set of stimulus materials, but it's resulted in tens of thousands of publications, so don't write it off. It's been okay for people. This is what that looks like. So this is one from Colin Davis. So in this case, the 2 words that we're interested in are stop and post. They're not, you'll forgive me that they're not phonological minimal pairs, but they're visual minimal pairs. And so this unit, which is the S unit, is most active in its connection to that word unit. So that means that this S is going to occur earlier in that word. And likewise, the S has a lesser activation in its connection to the other one, meaning that it will occur later in that word. But the letter units are shared. So in this case, the letter units are, it perceives a T as a T, but then its relationship to the word is mediated by an activation that says where in the word approximately should it occur, more or less to the beginning, more or less to the end. And then a recognition procedure for these things is just shown on the right. I won't go into any details. Really, we're just interested in the different data structure that was involved in coding for position in this case. So we've had so far a chaining thing and a thing that sort of says you can do this by coding by activation. Another thing that was hugely influential was Rick Ogren's model from the late 60s, where he did what he called contextualized letters, but they're better known, I think, at this point now as trigrams. So then for coding spot, we would say, you know, that you saw an S that was at the beginning of the word before a P and so on. So in this case, what we're really doing is mapping sequences to sets of trigrams or sets of bigrams or sets of some sort of thing. And this mapping is often not complete. So it means that at some point we will get collisions or aliasing, which you might be familiar with if you do signal processing, with longer forms, where longer things are then going to have the same representation as some shorter thing. So we can see this with this set of words, one of which is not English. Well, this one maybe isn't, although there's people's name, Bana. So in this case, this is the set of things that you get out of that. For trigrams, for banana, you get this one. But if we do banana, then you get the same one back. And if I did banana, we get the same one back. Now you can tell some differences between those things, but like the question is, at what, how are you doing and representing those differences? And in fact, if I give you banana and banana, then you might not be able to tell me same or different very well. Like, I don't know whether I did the same number in those two repetitions. Okay, this plays very strongly into the past tense debate because Rumelhart and McClelland chose this representation scheme to do the past tense calculations. And one of the criticisms that Pinker and Prince had about this was that it couldn't do the difference between banana and banana, right? So that is played out really strongly in the literature. So the most recent things in visual word recognition have been bigrams at a distance. So these are called open bigrams in the psychology literature, and the major article on this is Grainger and Whitney from 15 years ago. But then they've come up with they've been reinvented or whatever. So the Grainger's latest model, along with Jim Magnuson, who's at the University of Connecticut, they're calling them time-invariant string kernels, pulling on that literature from computer science now about string kernels. In DNA, this is called gapped K-mers. So like these are K-mer factors that are gaps, have gaps in between them. So that's another place where this has been used. Mori, who wrote a very influential book on machine learning, he calls them gappy bigrams. They're also called skipping bigrams or skipgrams. So you can just see by the explosion of terms for this that this concept has been rediscovered several times in the sort of related literatures. And so in this case, then spot and post or post would have representations where you would not only have the local sequences, but the sequences at a distance. So not only S comes before T, but S also comes before O and before P, right? And so we're representing these things. Now this looks redundant, but redundancy is not necessarily a bad thing. Like, so redundant codes are often more resistant to noise and lack of transfer in some cases. And so what does this look like? Well, so then this is taking from, essentially from Magnuson's paper. You can sort of see that the set of things that are being activated in stop is somewhat different than the things that are being activated in post, but they have some overlap too. And they do overlap in the unigrams in the sense of the individual letter units are the same in those two words, right? And so, but the individual unit letters being the same doesn't cause the two words to be indistinguishable. And then you can easily do these bigrams through just a two-way table, right? But just yes or no, adjacency matrix. From computational phonology, and this is heavily dependent on work that Jeff has done, we have the subregular hierarchy, which actually does exactly that separation into a successor function and a precedence function. So the goal in that work was to try to find phonotactic sound patterns in language. It wasn't so much about how are words stored in memory, but which patterns are people sensitive to when they're trying to say whether a word is possible or impossible in the language. And so at the strictly local level, you have those bigrams that are adjacent, and at the strictly piecewise level, you have the bigrams at a distance. So it turns out that this is another convergence between what was a word recognition theory and visual word recognition, and in fact what was phonotactic pattern recognition in computational phonology. And so the latest work has suggested that the information that you have in these and their complements is sufficient to characterize the phonotactic patterns that we see in the world's languages. So there's another convergence here between what was pattern recognition and what was storage of forms. So trying to put this all together, the desiderata that we have for this are, we want a static representation of a dynamic thing. So we want something that we can store in memory that doesn't play as a movie all the time. And this is very difficult. I mean, like, so in the case of a changing world, how do you store something in memory if it isn't just constantly changing? So how can we store things without storing everything as a movie when there's a change in there? And the other thing that we want to do in the case of most of these neural implementations is we're stuck with sort of a fixed length representation. We can't get new memory cells. We can't just sort of, and we can't actually put things in the memory cells. So we have to pre-calculate an awful lot of stuff. So we would like to handle sequences with repetitions. We'd like to be able to do concatenation, insertion, deletion, and so on. These should all be operations that we can do. And we'd like to have it be consistent with the empirical findings. And in fact, we'd like to have new experiments that would show that this is a better way to do this. So extending the work that I've done before and just grabbing these ideas from the CS literature, then we can have here a bag of sounds, that is the unigram representation, what letters compose the sound, what sounds compose the word. There's something special about initial and final. I don't really know what to do with this yet, but it's definitely there. And then the gap bigrams. And so on this case, we're going to do the recognition of a word, of incoming word, syllable by syllable. This is work that I'm ongoing with David Puppel and Nina Kazanova. So this would mean extracting the features from speech. And the best hypothesis about this that we have right now is that what you extract are spectrotemporal receptive fields, which look like chunks of a spectrogram. So I've just sort of done this. This is for Spot, actually, me saying Spot. And so then you would get these sort of areas outlined in red. This is not everything you get, but like this is a subset of the things that you get, which are then calculated by the auditory system. And the auditory system, this is not just for speech, it does this for every sound. So, and then the arrangement of the spectrotemporal receptive fields in cortex looks to be quite lawful and looks to be grown, basically. So that you have different tunings for rate and space and so on. And so this is very similar to receptive fields in cortex generally. So you have those things come in, and then what you need to do is to run a clock. The clock is driven by theta band oscillations by hypothesis. So these are the syllable level oscillations that are coming in, and then you drive a clock that does the internal gamma band calculations. So there's a theta gamma nesting in the endogenous oscillatory patterns of the brain. And so in this case, we're just going round in this circle again and again, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4. So you can think of this as common time in a song, if you'd like, 1, 2, 3, 4, 1, 2, 3, 4. And then we're getting the spectrotemporal receptive fields, and they're just firing when they're firing because like the outside speech has whatever temporal structure it has. And then what we're doing is we're looking for the coincidence detection of those things, just like in the barn owl. So we're the coincidence of the clock now with that thing. So we can say, we heard feature F at time 2, right? And so on. So now we need to carry this information forward in time so that we can collate everything that we heard inside the syllable to pass that on at the end of the syllable. Those things are represented in these boxes at the end. So this is what we heard at time 1 carried forward to time 4. So what we heard at time 1 carried forward to time 4. And what we heard at time 2 carried forward to time 4. What we heard at time 3, and so on. So these units at the outside, they'll be our interface into the word recognition system. And they represent the accumulation of information which may have come in at different times. So we might have information about the segment that's in the second position that came in at time 3. And so we're accumulating that information forward in time so that the output units are then our interface to the word recognition system. And then that calculation is first done to calculate the bigram pairs that are in there and the uniforms that are in there. And this is the circuit that you need to do that. So like we can pre-build this circuit and it will calculate what biphones were in the representation and what unigrams were in the uniphones were in the representation. And then those things taken together is the diagram that you saw before, where the competition between something like post and spot is in terms of the open bigram set and the uniform set that we've calculated from those other things. So now we have a hypothesis, at least, about how we go from spectrotemporal receptive fields to the activation of whole word units in the case of single syllables. Now, if you want multi-syllable words, which I'm sure you do, I do, then you'll need to face the problem of concatenation. And really, like, I think that that is the next big problem that we have to face. And I'll come back to that in a second. So what do we see here is really the patterned repetition of structure. We've got lots of similar units. We've got lots of similar circuits. The circuits are basically the same, except shifted one over. And you can grow that. And then, like, so there's lots of work in, like, B orientation and so on, which shows that, in fact, these circles of neurons and the columns associated with them grow in the B. Like, there's no, you don't need anything else. So, like, you can do this. We're carrying information forward in time. We're using a lot of disjunctive and conjunctive coding. And in this case, we're converting the sequences to sets. So, in fact, there is going to be collisions eventually. Although within the syllables, there are not a lot of, I mean, there are a few syllables that can have repeated sounds, but it's less than words. So we have lots of repeated neural assemblies, and like I said, these can grow these structures, actually. And so there is some supporting evidence for this model of unigrams and bigrams that we're proposing from birdsong. And so in this case, this was work I did in collaboration with Bob Dooling and Greg Ball, and our graduate student, Adam Fishbein. We had various species of birds. In this case, you're seeing the zebra finch motif. And so the original song is played at the front there, A, B, C, D, E, F. So there's 4 syllables is what the birdsong people call them. So those syllables. And then we have 3 conditions that we elaborate in various cases. One is just to turn the whole song backwards. Like, so it's like playing, you take the tape and reverse the whole thing. So this is just that thing flipped completely. Now, it's well known that the birds don't like this. So this is work that Tim Gentner had done. And so there's a particular song nucleus in songbirds called NCM. If you play the birds, the first thing, it drives NCM. NCM responds to it, you know, the neurons fire. If you play this thing to them, which is just the whole thing reversed, doesn't drive NCM. They just don't get any activity in the song center. Now, what else can we do? Well, we can have the in-between of the whole reversal. We can reverse the individual units. So in this case, we've reversed the syllables, right? So, you know, B is backwards. But it's still A, B, C, D, E, F, but the individual ones are backwards. And as opposed to that, we can keep the individual ones going in the right direction, but shuffle them around. So this is no longer A, B, C, D, E, F, it's C, A, D, B, and then you can ask, how well can they do this? Okay, this is the naturalistic thing, and then being crazy auditory scientists, we then create a perfectly controlled, spectrally modified version of this from Schroeder tones. And so, like, this is the controlled, you know, psychophysical analog of this. It doesn't really matter, the results are the same. So, like, you know, but like if you're, you know, if you're super interested in acoustics, then we can talk about that, but I doubt anybody here is. So here are the results that you get for this. And so the major things is a split between the 2 species, and we're still tracking this down a bit. But basically, the budgies can recognize when you change the background to the modified version all the time. So here is the whole reversal. They can recognize that you did this. This is done in a no-go, no-go pattern. Syllable reversals, they can do that. Reversing just the second syllable, they can detect that. Reversing just the second to last syllable, they can do that. And they can do the shuffled ones too, even though they're a little bit worse, especially at this one. So where you keep the first syllable the same and you shuffle the rest of the sequence. That is hard for the birds generally, because they must have something that says, this first syllable of this song is this syllable, right? And so this is causing an interference in this case, right? So if we shuffle everything, then they're better at it. And you can see that that's true in the other bird species too. We have two other species, the finch and the canaries, and it's easiest to see in the finch is the reversals they're quite good at. So this is where we take one syllable or more than one syllable and turn it around. But they're not good when we do the shuffling of the syllables. They're not good at detecting that. And the canaries are very much like the finch. The canaries are in here because the canaries display a different sort of plasticity. Finch learn their song once when they're juveniles, and that's the same for the rest of their lives. Budgies can learn new songs at any time. These are like, parakeets and so on, so you can like have them in your house, or parrots and so on. So you can have them in your house and you can teach them new things, like nobody expects the Spanish Inquisition. So Disco the Parakeet on YouTube. And then canaries are in between these because they learn a song in each year in relation to their mating season. So these are hormonally controlled. So in theory, now at the moment that they're tested, they're not in mating season. So they're held in our lab, not in mating season, and you need to perfuse them with a certain hormone to cause them to go into that. So, so they should look like the finch in the sense that they're not plastic at this moment, and they do. So one question that we get every time is like, how about doing that and just causing the canaries to become plastic again? There are technical reasons that that's difficult, so we haven't done that yet. But we will. And so we see the split between the two species. And then you can dive into this in greater detail if you'd like. So here are the unfamiliar songs, the familiar songs, and then the bird's own song manipulated in these various ways. It basically recapitulates the whole thing. Here we have it with the Schroeder syllables. So these are the perfectly controlled acoustic syllables. You get an even stronger effect. These are with the finch. So they can do the reversals. They can't do the shuffles. Same thing, And so overall, the take-home point is this, basically, is that in collapsing across all the different ones we have, here are the reversals, syllable reversals. They're quite good at it. When you shuffle the syllables, they're not good at it at all, well below chance. And the same thing is true with the canaries. And the budgies show a diminution, which is certainly significant, in ability when you have the sequences, but they don't lose that ability completely. And so our conclusions in that case is that the budgies are sensitive to the syllable order and the finch are not. And how we implement that, we think, is that the finch have a bag of syllables, like a grocery list, and to recognize their song, they just sort of say, was everything in there? And if everything was in there, that's okay. And it doesn't need to be in the right order. Now, in fact, they sing it in the right order all the time, so there's a puzzle there. The motor system has a fixed representation of the order that works, and somehow the perceptual system is not the same as the production system. So, there is a disconnect here between the perceptual system in non-plastic adult birds and the motor system. And so our approach at this point is now looking for neurons in the secondary or the auditory areas, area X, that selectively respond to sequences of A, B, or A, and then some other syllables, and then B. So we're looking at, to try to find this. Now, much to my objection, we're looking in the finch first, where we're not going to find this by hypothesis. So, so we really should have, in my opinion, started with the budgies, but again, for technical reasons, and I'm not doing the bird surgery, so I don't get to say this. So I don't get to dictate what surgeries they do. They've started with the finch instead of the budgies. Okay, so the conclusions are that the human animal brain representations for sequences are probably not like computer science strings. So it's probably not the case that we just have memory locations that we can just dump anything we want to in there, pull it out, change it, and so on. They're much more like pre-calculating what we could have seen. The other thing, that is maybe, difficult to come to terms with is that evolution generally builds composite solutions. And so really what we're seeing in the budgies is that they have what the finch have, plus they have something else. Like, so you don't throw away what works. In fact, you don't fix what is broke. Like, so it's broke to have just unigram representations for sequences, but they keep that anyway. Like, so, they haven't fixed it. They just, like, sort of added a patch, as it were. And so, like, this is bad computer science, but, like, maybe it's good evolution. And so we do see the different evolutionary states in the birds. So we see, that the finch, the idea here is that the finch are, if we rolled back the history for the budgies, they would have been like finch at some point. Do that. And so this is really a convergence toward a weird but simple, weird from our perspective, but simple data structure, which is this bag of sounds and gap bigrams. But it turns out that, like, that's converging from a number of sources. We do have a plausible set of conjunctive-disjunctive codings that we can do for this, so we can write a circuit down. And now the idea is that having been able to write that circuit down just like Jeffress did, we can go and try and look for that circuit. So now we know what we're looking for in terms of the connections between neurons in the birds. We can go do single unit recordings in birds. We can't do single unit recordings in humans. And then I think that what we're going to do is to find sequences and concatenations. So we're looking for sequences at the moment. Once we find those, then we would look for the operation of concatenation, and only then, will we be able to look for merge. Which is the fundamental operation that puts words together into sentences. So, thank you.
Hey, we have about 10 minutes for questions, but I'll let Bill go ahead and call your own questions.
If I can see you.
If he can.
So I might
Make sure you wave.
Yeah. Big motion. Motion is easier to detect than...
So about merge.
I only said at the very end.
No, so I think you gave us a very good overview of kind of like why this is an interesting problem from a computational perspective and how the implementation level is very interesting and intriguing. But I mean, for example, I'm a guy who's very firm at the computational level, and I'm already appalled when you try to drag me down to the algorithmic one. So kind of like, what is kind of like, from my perspective, what can I get out of having a better understanding of the implementation level?
Well, I think for you, you just need to wait. Like, and it might, outlast you, by the way. Like, so, because like, I'm not expecting to finish some of this stuff, by the way. If it took 40 years from Jeffress to Carr, like, then now I've done essentially now what Jeffress did, right? And, so it might be 40 years or more from now. I'm 56, I'm 96 maybe, but like, maybe I'll be alive then. But medicine does get better. But I'm not so sure that, that you'll see this. Now, I really think that this difference is like crucial, and you pointed this out. Like, so, this is sort of the operation, the fundamental operation you have in strings, and this is the fundamental operation you have in trees. Now, we have animal models for this, I think. Like, so we can go in and poke at the birds all we'd like. I don't think we have any animal models for this. Right, so, so like I think that looking for this on a neurocomputational level is, in my opinion, predicated on finding this. Because I think that this thing is a mutation of this. But, so like, from your standpoint, you can talk to me and like, encourage me and, but like, I'm not sure that I'm going to be able to get here. And you can be skeptical about people who said that they've got there. At least at the level of like, what's the circuit that does this?
Bigger.
Oh, way bigger.
Yeah. It's a question out of just curiosity, so it's pretty silly. So when you were showing us the bird songs, you showed that you sometimes flip the whole sequence and sometimes flip the syllable.
Yeah.
Does it make sense to do something where instead of, reversing the syllable, we would flip it? So like, let's say if we were looking at birds oo, where we would expect to see like...
You want to do spectral rotation?
Flip. Yes. So like if
well, the birds will just do that. We already know this. So like, there's a lot that's known about like bird auditory system. So if you do spectral rotation in the birds, they'll notice that.
Okay, so basically like rotation would give us same pattern with new elements.
Well, yeah, I mean, so what you're doing here is this is the background is playing, it's the same different experiment, modified to the bird. So this thing is playing in the background, and then at some point, it's like it's also like a habituation study with infants. So you play this in the background, and then you change it. And then the birds have to respond to the change. They have to go on a change and no go on no change. So you play this over and over and over again. And then at some point you either put this in or this in or this in. Now so what they're trying to do is detect, was that the same thing still or not? Was it something new? If we do the spectral rotation, we already know they'll do it because we know that they can hear that. Like we know for sure that they can hear that. Like this is the one that, actually, nobody thought that they couldn't do this, but they can't, right? Like this was the big surprise, was that, that when you broke the order of the things, they didn't care about it, right? Like, so, this was hugely surprising. And like the person that we have doing the neurophysiology doesn't believe this. Like, I mean, despite the fact that there's like a ton of behavioral evidence, he still just does not believe this. Like he says, of course the animals can do this. But they can't actually. Like, so like, I mean, we don't have, so the thing is that like you need to find something auditorily that they can't do. Like, so, but finding things auditorily that they can do, we can like multiply that endlessly.
And like a follow-up, so does it, like when we're reversing or flipping, we are creating new elements in the vocabulary of the bird?
Not in this case.
Not in this case, yeah. But does it mean that we are able to perform all those operations with the bird syllables and they're still recognized? Does it mean that birds don't have a finite alphabet?
No, they don't have a finite alphabet in that sense. They can, like, so when you put, well, that is a complicated question with the finch, because the finch learn their song only in infancy and then sing it that way for the rest of their life. And so actually not all that much, I mean, the vast majority of birdsong work has been on production of birdsong. And so the perception of birdsong like this, people just generally thought, well, they can recognize their own song, which they can. So then they thought, well, then that's, then they must be able to do like sequences. But actually nobody had tested it. And this was just thrown in. Like, this was another serendipity thing. So like this was just thrown into an experiment and they couldn't do it. Like, what's up with that? Like, so, so these cases, it's true that these are like out of vocabulary elements too. Like, so you can see it that way. Now, in the case of budgies, when you put them together in a new coop, they will negotiate a new song. And those new songs will include elements from both groups. So their lexicon of syllables is extensible in that sense. So the budgies are the only ones that I can speak to because, like with finch, you can't do that. And they don't really have a context song.
Thank you.
Yeah.
Is there like some window of sequences where this breaks down? So like if you have these random shuffled things.
They can't do these.
At all. Like
right.
Even at like a smaller sequence.
Well, if you make the cuts inside the syllables, then you're essentially doing something that's very much like this. And they can do that because they have like exquisite fine structure perceptual abilities. So like if you change each syllable, if you change any syllable in practically any way, changes that you would not be able to detect, the birds are able to detect those changes in the syllables. Like, so they can do spectral rotation, they can do reversal, they can do phase shift, they can do all sorts of things that we can't do. And so these are then controlled on all those things. So like, so that we can get this, so they can't. So like with these, you could do that. You could splice them internally. They wouldn't be able to do those because like there actually wouldn't be any difference in those things. And you wouldn't be able to do it either.
So would they like end up, if you keep playing these random shuffle things repeatedly, would they end up learning the pattern?
No, not so far. And like you're talking tens of thousands of trials. So like, I mean, maybe if you did 1 million trials, they would. But like, I'm not interested in doing that experiment. And two, like, I don't think that tells us much about humans, like at that point. Like, so if the training required is that intensive, like, whereas this thing we can run, on this one, we can run 2 birds a day. because like, you know, they can do that natural background. They don't need any long exposure to that. Like 100 is more than enough, right? So that looks much more like, you know, sort of general, like that kind of learning is not like the kind of learning that the starlings were doing when it took hundreds of thousands of trials before they learned something, right? And so this one is very rapid. You can do 2 birds a day. Yeah.
So on your wiring diagram, you had the mapping between form to the open bigrams.
Yeah.
Big one. A little bit backward. You had a gamma loop. I think it was a couple of slides before this, but it doesn't matter.
Yeah.
A gamma loop, but I always thought that this one. Yeah. So I always, or sorry, it's a theta loop, whatever.
So yeah, theta. I mean, the hypothesis, although this is not what general neuroscience thinks.
Right.
So the hypothesis is that theta is driving gamma. That gamma is nested inside theta.
That's right.
And so that the start of a new syllable, which is indicated by being someplace in a particular phase, like Gitsa thinks it's at the top of the phase. So in theta, band is then going to drive gamma band. The general theory in neuroscience is the opposite, is that gamma band drives theta band.
So is there any interaction here between the theta and gamma bands, or is it just implicit and it doesn't matter for this slide?
Well, for this slide, I don't care. But yes, I mean, like, I think that, you know, if somehow this computation overall results in a lack of coincidence, then that's a signal that the theta cycle is misaligned. But, you know, what that kind of feedback would be, I don't know. And I don't know how to assess, really, at this point, how well, you know, the coincidence detection worked. So like at this point, it just is doing what it's doing. I mean, so there's no evaluation of how good a coincidence detection that was.
I think we have time for one more question, then we'll...
Yeah.
So you maintain the relationship between the spectral and the temporal properties in each syllable, is that right? Is there any work sort of manipulating those independently?
What would you like to do?
So I'm thinking about, you know, changing the time course of the spectral properties.
I see. Yeah. So you know so if you change the time course, that's essentially like dynamic time warping, and so that there are 2 views on that. So one would be that I'm slowing the clock down. So like in that case, if all I'm doing is slowing the clock down, then fine. Like, you know, it'll just work the same and just work on a slower clock. So like, you know, that would be modulated.
The time is a crucial cue for particular,
well, you can,
as in like voicing.
So there's only a factor of 2 here, right? So like, you know, it's not a factor of 100. So like you can make the things twice the length and it'll still be intelligible. You can shorten them by about 2 times, make them half the length and they're still intelligible. Go outside those bounds, it's no longer intelligible. So it seems to be strongly related to the bounds on theta and gamma oscillations in the brain. So if you're too far outside the endogenous oscillation regime, you can't match it.
Right, but I'm thinking of, you know, just changing the relative timing between, you know, this spectral cue and the following spectral cue.
Right. So the relative timing, you know, if you want to destroy the relative timing, right, then you will get a different percept in most cases.
Right, in speech, I'm wondering in bird, you know, so has anybody done anything like that with birds?
Well, in speech we do it all the time, but then like it's a different word. Like, you know what I'm like, so like lots of our things manipulate that. In the birds, the problem with the birds is you can't ask them, did they hear spot or stop? You can only say, did you hear the same thing again?
Right.
So all you'll get is, no,
So
I didn't hear the same thing again.
How much can you mess up the timing before you get them not?
Oh,
you know,
about 20 milliseconds.
Yeah, okay.
Okay. So I guess there was an easy answer to your question, but I wasn't sure what you're asking.
Okay, let's thank Bill again.